started. Let's start the program.
Sure.
Hello, everyone. I welcome you all for the webinar on cancer strategies, which is brought to you by the Biomix team. Today, we have one of the leading scientists in oncology from USA, Dr. Srikumar Chellappan, who is currently working as a professor and chair in the Department of Tumor Biology and a senior member in Cancer Biology and Evolution Program at Huxley Motives Cancer Center and Research Institute in USA. I thank Dr. Srikumar Chellappan for accepting my invitation and making a joyous presence over here with us today. Our speaker will reach you all with this talk on targeting the untargetable novel strategies to combat cancer. And his talk will be followed by question and answer session. So participants can place your questions in the chat box of YouTube. And I am much delighted to introduce our speaker today, Dr. Srikumar Chellappan, who is currently working as a professor and chair in Department of Tumor Biology and a senior member in Cancer Biology and Evolution Program at Huxley Motive Cancer Center and Research Institute in USA. He was a postdoctoral fellow at the Department of Microbiology in Howard Hughes Medical Institute, that University Medical Center in North Carolina. He obtained his PhD in biochemistry from Indian Institute of Science, Bangalore, India. He obtained his BSc and MSc in zoology from University of Kerala, India. Dr. Srikumar Chellappan has held various reputed positions right from 1991 to 1992. He was a research assistant professor in the Department of Medicine at University Medical Center in North Carolina. From 1992 to 1999, assistant professor in the Department of Pathology in the College of Physicians and Surgeons in the Columbia University, New York. From 1999 to 2001, he was the assistant professor of clinical pathology in College of Physicians and Surgeons in Columbia University, New York. From 2001 to 2006, he was the associate professor in drug discovery program in the Department of Interdisciplinary Oncology at Huxley Motive Cancer Research Center Institute, Florida, USA. From 2006 to 2010, he was a senior member drug discovery program at Huxley Motive Cancer Center and Research Institute, Florida. At the same time, he was also a professor in the Department of Oncology Sciences, University of South Florida. And he was also a professor at the same time in the Department of Molecular Medicine in University of South Florida, USA. In 2011 to 2014, he was a scientific director at National Functional Genomics Center, Motives Cancer Center. Since 2010, he is being a chair of Department of Tumor Biology, Motives Cancer Center, and from 2015, he is being a Motives Distinguished Scholar. Dr. Srikumar Chellappan research interest falls in signal transduction, transcriptional control, and cell cycle regulation, nicotine receptor signaling, and lung cancer and cancer stem cells, and also in angiogenesis and metastasis and drug discovery. He is a recipient of several awards and medals such as Dr. N.G. Guru Memorial Gold Medal and Dr. K. Bhaskaren Nair Gold Medal in 1981 for obtaining the first rank in the University for MSc. Dr. K.V. Giri Memorial Gold Medal for the best PhD thesis in the Department of Biochemistry in IISc in 1987. Huxley Motive Cancer Research Center Scientist of the Year in 2007. Winner award for award in Motive of American Asian Scientist in Cancer Research in 2016. He holds membership in many scientific societies such as American Association for Cancer Research from 2002 still present, American Society for Microbiology from 1998 to 2015, New York Academy of Sciences from 2006 to 2012, Association for Biochemistry and Molecular Biology from 2006 to 2015, DMT International Association from 2009 to 2012, Metastasis Research Society from 2013 to 2016, Society for Research on Nicotine and Tobacco from 2013 to 2015. Dr. Srikumar Chellappan has 5 US patents so far, and he was an invited speaker in various reputed scientific forums internationally. He is an editorial board member in various reputed journals such as editorial board member in molecular pharmacology in 2007 to 2014, associate editor in molecular pharmacology in 2014 to 2016, associate editor in molecular cancer, which holds the impact factor of 15.3 in 2010 still now. Associate editor of Neoplasia in 2017 from 2017 still now. Associate editor in Cancer Cell International from 2017 to 2018, editorial board member in the Journal of Oncology from 2012 to 2018, and academic editor in PLOS once from Feb 2012 till current date. Such an eminent resource person is with us in our Bionics platform today. With this very brief introduction, I request Dr. Srikumar Chellappan sir for his talk.
Thank you, Partha, for the very kind and generous introduction. Thank you so much. Thank you for the introduction as well as for the invitation to present the seminar to the students and postdocs and other trainees in India. It is always a pleasure to present our work and give a background of the type of things we are doing, as well as some of the strategies that people are using nowadays to combat cancer. What is the issue? What are the main problems in curing cancer? And stuff like that. And I will also present a significant amount of the work that we are doing in our lab. So let me set my timer to 55 minutes and I will start sharing the screen with your permission.
Yeah, sure, sir. You can proceed.
Sure. So just 1 second. Spotlight. Yes. Okay. So the cancer incidence in the US and India, it is a, as you know, let me go back to the previous slide. Yes. So it is a major problem in all the countries across the world. And as you know, it is one of the leading causes of disease related mortalities in all over the world. Something like 1.89 million projected new cases will be detected in 2021 in the US. Some 600,000, 6 lakh people will die from cancer in 2021 in the US. At the same time, it has been promising some of the advances we had made. There has been a 31% reduction in cancer-related deaths from 1991 through 2018. To a great extent, this is because of the cancer prevention effort, especially the reduction in smoking and good lifestyles and healthy eating, etc. At the same time, there also has been a significant increase in the development of novel therapeutic strategies, the development of new drugs, the development of immunotherapy, etc., which is causing a reduction in cancer-related deaths all around the world. Generally, risk of developing cancer in a lifetime for any person, it is 42% in males, so 42% of the males may get cancer before they die, 37% in females. Risk of dying cancer from cancer is 22% in males and 19% in females. These are information we get from the Siegel et al. Paper, which says cancer statistics, and it is published in January or February of every year. As far as India is concerned, there is 1,392,000 patients who are estimated to be living with cancer in India in 2020. And the cancer incidence in India is appearing to be increasing. There is a marked variation in the incidence rate across India, with a remarkably high incidence of cancer in the Northeast states and certain cities in the Northeast. So the major sites of cancer in India are breast, lung, mouth, cervix, uteri, and tongue. So in India also, now there is a publication where they have been publishing or they have been conducting statistical analysis of the cancer incidence and cancer deaths all across the country in population-based studies, as well as data they get from hospitals. So they publish that every year, just like in the US, starting recently. So one of the major reasons why cancer therapies fail, this is an example that is shown here, is the development of resistance. So this is a very historical figure which I published around 10 years ago, where here is a person with melanoma. There are, you can see the tumors all over his body. They were given this person was given this particular drug, PLX4032, which is an inhibitor of a kinase called BRAF, mutant BRAF. After 15 weeks, he was completely cured. But within 23 weeks, the tumors reappeared and it became very bad. And of course, he did not survive this. So this is a very common phenomenon in cancer. If you are treated with a specific targeted inhibitor of a kinase or a specific protein, invariably people develop resistance. So this is one of the major reasons why the cancer mortality is so high. So overcoming drug resistance and how to combat this phenomenon is a major challenge remaining in cancer. So other challenges that remaining. So cancer is not just a group of cells or just one type of cell sitting together as an organ or as an organoid and growing. So if you take a cancer and look at it, you find all these cancer cells shown in gray. There are cancer cells which migrate and metastasize or spread to other organs. In addition, the cancers also contain cells and inflammatory cells, essentially B cells and T cells, tumor infiltrating macrophages, tumor infiltrating lymphocytes. There's a large amount of inflammatory cells in the cancer. In addition, there are pericytes, there are cancer-associated fibroblasts, there are endothelial cells and vasculature, which cause angiogenesis and provide supply to the cancer. So cancer is not just one type of cell. So if you say that someone has lung cancer, for example, or skin cancer, it is not just a group of skin cells or abnormal lung cells sitting around. There's a whole bunch of cells contributing to the tumor. So they all support each other. The cancer, the pericytes or the fibroblasts, they secrete multiple growth factors and cytokines that facilitate the proliferation and invasion and migration of the cancer cells. The inflammatory cells, for example, the immune cells, many of them can kill the cancer cells, but many of them are rendered inactive or inefficient in killing cancer cells. And they also produce cytokines and provide an inflammatory environment facilitating the growth of the tumors. So curing cancer is not just killing these particular cancer cells. That itself is challenging in a tumor, but given the complexities of the tumor microenvironment, it is pretty challenging. So these are some of the problems by which the cancer cure rates remain to be fairly low, even though it is improving due to multiple reasons, some of which I will touch on later. So one of the type of cells present in a cancer in the past 20 years or so, people have proposed that there is a subset of cells within a cancer called as a cancer stem cells or cancer stem-like cells. So classically, it is believed that every cancer cell in the body or in a tumor can divide. Of course, they divide, and they acquire mutations or changes in their genetic makeup. Oncogenes are activated, tumor suppressor genes are mutated. The tumor suppressor genes are inactivated, whereas oncogenes are activated by mutations, exposure to carcinogens. So the classic hypothesis was that every single cancer cell has the potential to initiate cancer and to propagate cancer. Here it is acquiring more mutation, it can form tumor again, it divides into cancer cells, again it can form tumor, etc. So every single cell, according to the old model, had the ability to form tumors. But recently, based on studies from leukemias and from hematological malignancies, people found that there is a subset of cells within a tumor, or only those particular subsets can initiate and propagate tumors. They call it as cancer stem cells. So a cancer stem cell is a schematic of a cancer stem cell. There may be 1% of cells or less than that in a tumor or cancer stem cells. They sit around like this. So they acquire mutations, or the normal adult stem cells acquire mutations. The cancer stem cells now divide in an asymmetric fashion. It divides into another cancer stem cell or a more differentiated non-stem cell. And these particular cancer stem cells are the only cells that can give rise to a tumor. So they give rise to additional cancer stem cells, which again give rise to a non-stem cell and a stem cell, and the stem cells keep on producing the tumors, whereas the non-stem cell, when it divides into a stem cell and a non-stem cell, these non-cancer stem cells cannot form tumors. They just sit around and they contribute to the bulk of the tumors. So how did they find this? They took human tumors or mouse tumors, they dissociated into single cell suspensions and put it back in mice, immunocompromised mice. And they found that only about 1% of the cells could cause tumors in mice. And those tumors also they dissociated, put it back into another group of mice. Like serial transplantation of these tumors, they found that only a very small percentage of the tumor cells can form or can initiate and propagate tumors. So based on that, they came up with this hypothesis of the cancer stem cell hypothesis. So these are identified based on um, multiple features, cell surface markers. For example, breast cancer, there are certain markers called CD44, which is expressed on the cancer cell, high levels of CD44, low levels of CD24, high levels of ALDH1, etc. For each cancer type, there are cell surface markers which have been identified, which characterize these stem-like cells. So my talk will be mainly on cancer, on lung cancer, and the role of cancer stem cells in the process. And so what are the properties of the cancer stem cells? They can self-renew and divide asymmetrically. As I mentioned, they can give rise to more cancer stem cells and non-stem cells. They are very slow dividing cells that can initiate tumors. So the problem of the slow division is that they can remain dormant in the body for a long time. For example, many of the breast cancer patients who are completely cured of cancer by chemotherapy, they will develop cancers again after 10 or 15 years. And those cancers pretty much have the same phenotype as the original cancers. That is probably because there are these dormant cancer cells sitting in the bone marrow as well as other organs. And when they revive, they cause cancer. So it is mainly these stem-like cells that remain dormant and that are slow dividing that is causing this. The slow division also causes some other problems. Many of our anti-cancer drugs target the cell cycle machinery and the DNA replication machinery. So if these cells are very slow dividing and they're not cycling, these drugs will be inactive. So that's another problem with this. So they're able to generate heterogeneous tumors. I will show you one bit of data generated by Sandeep Singh in the lab, who is going to give a seminar tomorrow in the same venue. I strongly encourage you to listen to his talk. So what he and others found was that the two cancer cells, the stem-like cells, can give rise to multiple cell types within a tumor. So I will show you a little bit later. For example, some of the angiogenic phenotypes, the growth of the blood vessels, can be facilitated by these stem-like cells. So the stem-like cells can remain dormant for a long time, and they can revive back depending on the changes in the physiology of the body. They are highly drug resistant. The reason for the drug resistance, we'll come to it a little bit later. They overexpress a lot of drug exporters. They overexpress a lot of survival proteins. They cause certain changes in the cancer cells itself that facilitate their resistance to the drugs. They are multipotent, as I mentioned. They can differentiate into multiple cell types in vitro. They have different metabolic, microRNA, and epigenetic signatures, and they're generally identified and isolated based on cell surface markers or drug exclusion. So will be talking about lung cancer mainly, though many of the stuff I'm going to say is valid for other tumor types too. So one of the reasons why we have been working on lung cancer is that lung cancer is a cancer that causes a maximum number of mortality back here in the US as well as back in India. And there are tobacco-specific carcinogens. They are associated with 90% of small cell lung cancer and 60% of non-small cell lung cancer. So these are the two major groups of lung cancer: small cell lung cancer and non-small cell lung cancer. The small cell lung cancer is generally neuroendocrine type. They are very small cells, fairly difficult to culture. They respond very well to chemotherapy and nowadays to immunotherapy. And the chemotherapy tumor who respond to chemotherapy invariably develop resistance after some time. So the mortality rate is very high. Non-small cell lung cancer, mainly adenocarcinoma, squamous cell carcinoma, large cell carcinomas, they can be treated by multiple methods. I will touch on that a little bit later. So traditionally, tobacco carcinogens like NNK and NNN, they form adducts with DNA and mutate vital genes like Kras, P53, and RB. So these are the genes, as you know, Kras is a major oncogene, P53 and RB are major tumor suppressor genes. These are mutated by these tobacco carcinogens causing cancer. And our studies, as well as many other studies, showed that the major addictive component of the tobacco smoke, nicotine, they may function in a receptor-mediated fashion to induce cell proliferation. So nicotine is the one that causes addiction to tobacco, and that's probably one of the most addictive compounds in the world. So nicotine by itself is not carcinogenic, but it can promote cell proliferation, angiogenesis, and inhibit apoptosis. So it is mainly a tumor promoter. So once a tumor is initiated, it can promote the growth and metastasis of tumors. Hope I am making myself clear. So this was an experiment that was done by one of my earlier students, Becky Kinkard, in the lab. What she did was that she implanted tumors onto the skin of mice, uh, line 1 cells onto the skin of the mice, and she gave nicotine, 1 milligram per kilogram nicotine to the mice for around 18 days. And what we find is that the mice receiving the nicotine had significantly larger tumors, as can be shown here graphically. So as you know, people don't take nicotine by injections. They use electronic cigarettes. They use nicotine patches, which I think is very popular in India also now, nicotine gum, etc. So what Becky did was that she implanted tumors under the skin of the mice and put nicotine patches on the mice. And she found that the tumors are significantly larger as well. And not only that, these tumors eventually metastasized. And when we look at the spreading of the cancer, we find that the mice exposed to nicotine had significantly more metastasis compared to the control or the vehicle treated mice. So one of the major features of metastasis, one of the major events that facilitate metastasis is this phenomenon called EMT, or um, epithelial mesenchymal transition. So as you can imagine, most of the cells of the body, for example, if you look at your skin cells or the cells inside the duct of the mammary duct or the lung alveoli, what you find is that they are very nicely arranged like the tiles on the floor with some nice grout or cement holding it together. These are the tight junctions. This is a cell line. So what we are finding, if we serum starve them, they may do some of the tight junctions. But if you treat these cells with nicotine, what we are finding is that these cells lose their attachment to each other. These flat, tightly packed cells become very spindle shaped. They become almost like fibroblasts or very mesenchymal in nature, hardly any tight junctions. They, they become very spindle shaped, and now they are free to move about, spread to other sites, and metastasize. So these cells enter the bloodstream by intravasation, go through the bloodstream, land in other organs, settle down there, and grow. So this process is called EMT, and we found that EMT is significantly induced by exposure to nicotine. Okay, so not only that, as I mentioned earlier, the stem-like cells can self-renew. They can produce more stem-like cells. And you can measure that by a sphere formation assay. Again, it was established in the lab by Sandeep Singh when he was working here at Moffitt a dozen years ago. So what we found after that was that if you take the stem-like cells based, this is SP cells, or stem-like cells based on a cell surface marker, these control cells, they form very small spheres. Now, if you take the stem-like cells, treat them with 1 micromolar nicotine, you get a huge growth or proliferation of these cancer stem-like cells. If they self-renew, produce more stem-like cells. This is from one cell line. This is a non-stem-like cell. You don't get that. This is a stem-like cell from another cell line, divides very well, form the spheres. And not only that, these are extracts from electronic cigarettes. Now, e-cigarettes are very popular, and they are being pushed as an alternate to smoking. And what you are finding is that these can also essentially induce the cell proliferation of this particular stem-like cells on one cell line, another cell line. So nicotine can not only induce EMT, it can also induce the self-renewal of the stem-like cells. Because of these findings, we were very interested in understanding the biology of the stem-like cells. And I think my next few slides will be dealing with the stem-like cells. What is the basics of this? What is the biology of this? And why is it important? So if you remember what I said earlier, the features of the stem-like cells, they are the cells that initiate the tumors, propagate the tumors, they are highly drug resistant. So you can imagine a person being treated for cancer. The drugs will eliminate the non-stem-like cells, but the stem-like cells are still present in the body, and they will produce more stem cells. The cancers will continue to grow. So unless you eliminate the stem-like cells or the cancer stem cells and the non-stem cells, it will be very difficult to completely cure cancer. So it is absolutely essential to understand the biology of these cancer stem cells and to devise strategies to target them and to eliminate them. So some of the studies by Sandeep Singh, who again, as I mentioned, who is going to talk tomorrow in this venue, he is studying the genomics of cancer-associated fibroblasts. And Namrata Bora Singhal, who is an IAC alumni. What we found was that there is this particular, these are three transcription factors that are necessary for stemness: SOX2, OCT4, and NANOG. What we found was that it is this particular transcription factor, SOX2, that is absolutely necessary for this lung adenocarcinoma stem-like cells to self-renew and form more stem cells. So here cells transferring the control siRNA. When you deplete SOX2, um, the tumor growth is the self-renewal is significantly reduced. When you deplete these two, there's much less effect. So SOX2 is absolutely critical for the self-renewal and the maintenance of the stem-like cell population in this particular cancer type. So what we found was that, again, this was work by Courtney Schall. What we found was that if you take cell lines and you expose them to nicotine or to e-cigarettes, you get a very good, very fantastic induction of notable marked induction of SOX2 protein by nicotine or with the e-cigarettes. And this is happening at the transcriptional level. So SOX2 is induced by nicotine as well as by e-cigarettes at the transcriptional level. So the question is that how is SOX2 is induced? So our studies, which were conducted by Namrata Bora Singhal, what we found was that we did a differential gene expression analysis of proteins that are differentially expressed in the stem-like cells versus a non-stem-like cells. What Namrata found was that there was this various proteins are differentially expressed, but this particular protein, YAP1, this stands for S-associated protein. Uh, so this particular protein is definitely overexpressed in the stem-like cells. So what is a S-associated protein? It is a protein that is an oncogenic component of the hyposignaling pathway. So what is the hyposignaling pathway? It is a pathway that essentially regulates organ size. When the cells are tightly packed, when the organ has reached the proper size, this pathway gets activated. So how does it get activated? By cell-cell contact, the tight packing of cells. It activates these two proteins called expanded and Merlin, which activates MST1 and MST2 kinases, which activates another kinase called LATS1 and LATS2. And this LATS1 and LATS2 protein phosphorylates the YAP1 protein or TAS protein, which is a very closely related protein, leads to a degradation, and this oncogenic protein is eliminated. But when the cells are very loosely packed, or when the cell or when the organ is poorly developed or developing, this pathway is inactive. So now there is no MST1, MST2 activation, no LATS1, LATS2 activation. So there is a lot of accumulation of YAP1. YAP1 goes into the nucleus and induce transcription of a variety of genes involved in cell proliferation, EMT, and metastasis, thus promoting tumor growth and metastasis. So when we found that such an oncogenic protein is being elevated in the stem-like cells, we examine whether that is involved in regulation of SOX2. Hope I am making sense and hope it is all logical. So what exactly is YAP1? It is a transcriptional coactivator. It essentially binds to other transcription factors and promotes transcription. It does not bind to DNA directly. It does not have any enzymatic domain. It gets phosphorylated and it gets degraded, etc. But essentially, it binds to other proteins and facilitates transcription. That is its major function. So why is it important? There's a large number of tumors where YAP1 is altered, gliomas, non-small cell lung cancer, which we are interested in, esophageal cancers, so many cancer types where YAP1 is overexpressed, amplified, or more retained in the nucleus. So a whole variety of cancers show aberrations in the YAP1 protein or increased levels of the YAP1 protein. So if you can target this particular protein, it will be very beneficial in eliminating the cancer stem-like cells, as well as for reducing the tumor growth, as well as metastasis, excuse me. So to show the more importance of this particular protein, when we look at tumor tissue microarrays, that is array containing so many tumors, we find that the YAP1 levels are significantly elevated in the tumor tissues compared to the normal tissues. And the tumors harboring higher levels of YAP1, the patients have a poor survival rate, whereas those harboring low levels of YAP1 have a higher survival rate. So YAP1 is definitely important for non-small cell lung cancer. So this is again some of the data which I mentioned a little bit earlier. We've Namrata found that YAP1 levels are higher in the stem-like cells here compared to the non-stem-like cells. And this is happening at the level of the transcription, the messenger level also. The YAP1 proteins are elevated in the stem-like cells compared to the non-stem-like cells, etc. And when you deplete YAP1, there is a complete suppression of self-renewal of the tumor, of the stem-like cells of the sphere formation assay, as seen in the spheres formation assay. So there is a self-renewal of the stem-like cells is completely eliminated when you deplete YAP1, two different siRNAs. And if you put these cells in mice, when you have parental cells implanted in mice, you get very good tumor growth, notable induction of tumor growth. And the cells that lacking YAP1, there's hardly any tumor growth as seen here. But we also know that it is happening through SOX2, as I showed a little bit earlier. So if you take these tumors and these cells which lack YAP1, and now you start overexpressing SOX2, you find that you get the tumor growth back. So these are cells with no YAP1, no tumor growth. Now to these cells, you add SOX2, the tumors come back, suggesting that the induction of SOX2 by YAP1 is one of the major ways by which YAP1 promotes stemness and perhaps tumor growth. Is YAP1 have any other relevance? Yes, it does. Because YAP1 levels are significantly induced by nicotine and e-cigarettes, as seen by an immunofluorescence experiment. So these are A549 cells and mesenchymal stem cells. Fair amount of YAPN, not a huge amount. You treat the cells with nicotine or e-cigarette extracts, you can find there's a marked induction of YAPN levels in the cytoplasm, but predominantly in the nucleus, showing that YAPN is also a target for nicotine and the tobacco carcinogens. So to switch gears a little bit, so whatever I showed so far, what I showed is that nicotine and tobacco smoke components, they promote the division or the self-renewal of cancer stem cells. This happens through this particular transcription factor, SOX2, and SOX2 is induced through this transcriptional coactivator, YAP1. So coming back to the cancer stem cell phenotype, I want to divert a little bit. Angiogenesis is a major feature of cancers. As you know, tumors that have to grow or that when they reach around 1 centimeter in diameter, the inside of the tumor becomes hypoxic, and they have very little access to oxygen or food from the normal blood supply. So the growth of new blood vessels or neoangiogenesis is absolutely essential for tumors to grow beyond 1 centimeter in diameter. So a whole variety of endothelial cells will come to the tumor, and the tumor cells will secrete proteins like the vascular endothelial growth factor, which attracts the endothelial cells, promote the innervation, formation of the blood vessels, and the nutrient supply as well as the oxygen supply is restored to the middle of the center of the tumors, and the tumors will continue to grow and expand. Mary Hendricks, a very renowned researcher around 25 years ago, noted that before the actual blood vessels starts to grow and innervate these tumors, a subset of the tumor cells by themselves can acquire this tubular phenotype and start providing nutrients and oxygen. She called it as vascular mimicry or vasculogenic mimicry. So essentially, a subset of the cancer cells are acquiring the tubular phenotype and starting the blood supply. And it has been shown after that some of the cancer stem cells are the ones that are doing this. There are some very nice papers in glioma, as well as some of our studies where we show that some of the cancer cells can really, the cancer stem cells can acquire this phenotype. So this is an experiment done by Sandeep long ago, where he took the cancer stem-like cells, put it in matrigel, treated them with VEGF. You get this very nice vascular network or vascular-like network. We know it is vascular-like because they express CD31, which is a marker for early endothelial cells. And so this is a pure cancer cell that, given the proper conditions, acquiring the phenotype of endothelial cells and acquiring this whole tubular phenotype. A non-stem-like cell cannot do that. So only the stem-like cells can do that. So this was very interesting, and we decided to examine what is the role of YAP1 in this whole particular phenomenon, and that has taken to a totally different direction. So that paper is under review, and hopefully it will come out in the next few months. So what we found was that YAP1 protein regulates the expression of VEGF, that's a vascular endothelial growth factor, which is mainly induced or secreted by cancer cells. What we are finding is that if you deplete YAP1 by siRNAs, there is a strong suppression of VEGF expression. The VEGF expression is significantly reduced by two different siRNAs in one cell line. This is another cell line. It goes from here to here. The VEGF expression and the YAP1 expression goes from there to there because of the siRNAs, suggesting that YAP1 is necessary for the expression of VEGF by the cancer cells. So this is again showing the same thing by shRNA, and we have the same data with sgRNA as well. So how does it happen? How does YAP1 do this? So there's this very well characterized protein called HIF1 alpha, hypoxia inducible factor alpha, which is known to bind to the VEGF promoter and induce its transcription. So the whole biology of VEGF and HIF1 and another protein called VHL was so important that Bill Kaelin and many others won the Nobel Prize for it 2 years ago for the study of the VHL protein, HIF1 alpha, and the whole hypoxic response. So this is an example where we find that this YAP1 protein physically interacts with this particular transcription factor. So this is a proximity ligation assay. This is almost like a double immunofluorescence. So each spot you are seeing is a foci of interaction between two proteins. Under normoxic conditions, when the cells are grown under conditions of normal oxygen, 5% oxygen, you find a certain amount of interaction in two different cell lines. But if you reduce the oxygen level to 1%, you can find there's a huge induction or the huge increase in the binding of YAP1 to HIF1 alpha protein. We are showing that by immunoprecipitation Western blots as well. So this shows that under hypoxic conditions, the YAP1 protein, the levels go up, it binds to HIF1 alpha, and perhaps promotes VEGF expression and angiogenesis. How do we know that? Yeah, so then this is another slide before that, where we are showing that the physical interaction of YAP1 with HIF1 alpha is elevated in lung cancer. So this is a normal lung. You can see one or two spots here and there. This is a squamous cell carcinoma of the lung. This is adenocarcinoma. This is metastatic carcinoma. In all these tumor types, we are finding there's an increase in the binding of YAP1 to HIF1 alpha in the tumors compared to the normal cells, suggesting that the increase in this physical interaction as well as the increase in the levels of these proteins has contributed to the oncogenesis. So how is HIF1 alpha regulated by hypoxia? How does HIF1 alpha levels go up in the hypoxic conditions? So under normoxic conditions, there's a protein called prolyl hydroxylase. So this is a protein that can acquire oxygen from the surroundings, from the cells, and add hydroxyl groups to prolines. So the prolyl hydroxylase to PHD2 under conditions of normal oxygen, it adds hydroxyl groups to the proline amino acids on HIF1 alpha. This leads to ubiquitination of this protein and VHL-mediated degradation. VHL is a ubiquitin ligase. So once protein gets ubiquitinated, it gets degraded. So when the oxygen levels are high, this prolyl hydroxylase, especially prolyl hydroxylase 2, keeps the HIF1 alpha levels very low in the cells. Under hypoxic conditions, there is very low oxygen. This enzyme is inactive. So now the HIF1 alpha protein can bind to the hypoxia responsible response elements like VEGF transcription growth factor and induce the transcription and induce all the necessary genes for angiogenesis under hypoxic conditions. Surprisingly, we found that a very similar regulation takes place in the case of YAP1 as well. So we find that YAP1 levels are prolyl hydroxylated under normal conditions. Under hypoxic condition, the prolyl hydroxylation is significantly reduced. And in vitro also, it can be hydroxylated by the hydroxylase enzyme. And we find that just like in the case of VHL, in the case of HIF1 alpha, YAP1 also binds to VHL under normoxic conditions, which keeps its level down. Under hypoxic conditions, there is no interaction with VHL. There is increased binding of YAP1 to HIF1 alpha, contributing to angiogenesis and cancer progression. So this was a quick summary of the angiogenic functions of YAP1. Under normal conditions of neoangiogenesis, this normoxic cells, there is PSD2 hydroxylating YAP1. It leads to the binding to VHL1 and degradation, whereas under hypoxic conditions, there is no VHL1 binding to YAP1, there is no hydroxylation. YAP goes into the nucleus, binds to HIF1 alpha and other transcription factors and induces transcription of the genes like VEGF, which promotes tumor growth. Again, showing one more reason how to suggest that why and how it would be important to target this YAP1 protein to combat cancer and to eliminate cancer stem cells. So how can we target cancer cell, the YAP1 protein? As I mentioned, YAP1 has no enzymatic activity, so you cannot design an inhibitor that prevents its enzyme activity. It does not bind to DNA. Anyway, transcription factors are difficult to target with small molecules anyway. So we try to devise other methods to target YAP1. So what Namrata found was that the YAP1 protein can be detected on this particular promoter region of SOX2, where a protein called Oct4 binds. And we found that YAP1 binds to Oct4 on the promoter. And if you do co-transfection experiments, if you overexpress YAP1 along with the SOX2 luciferase, we find that YAP1 can induce the promoter in a Oct4 dependent fashion. So YAP1 binds to another transcription factor called Oct4, and that is shown here. This is YAP1 stained with red, Oct4 stained with green. And if you overlap the images, you find very nice areas of overlap or colocalization. Sorry for that. And we also find map, Namrata also find map the region of YAP1 that binds to Oct4. We found that the WW domain of YAP1 binds to a PPX5 modif of Oct4. That is shown here in a little bit more detail. This is a WW1 domain, which is the Oct4 binding domain. So what she did was that, so our hypothesis was that if we can prevent the binding of Oct4 to YAP1, if we can prevent this interaction, can you suppress the expression of SOX2? Can you prevent SOX2 from being expressed and prevent the self-renewal of cancer stem cells? So what Namrata did was that Namrata synthesized a peptide corresponding to this region, attached it to a carrier peptide and delivered it to cells. And what she found was that when she did that, it could disrupt the binding of Oct4 to YAP1. See, there's a lot of interaction here. The interaction is reduced. When you do co-transfection experiments, when you put the peptide in, the expression of SOX2 gene is reduced. And when you put this peptide into cells, you find that the self-renewal is also significantly reduced. Just showing a small snippet of the data, suggesting that if you disrupt this binding, the Oct4-YAP1 interaction is abrogated. You can target stem-like cells using this means. So we devised a drug discovery strategy in the lab where we examined multiple drug libraries from the NIH. We also designed PPX5 mimetics, that again mimetics that correspond to the PPX5 domain of the Oct4 protein. We did in vitro binding assays, SOX2 luciferase assays. We examined the ability of these compounds to suppress self-renewal. We did a whole amount of studies, and we have identified some small molecules that essentially disrupt the binding of Oct4 to YAP1. So this is an example of some of the compounds where each, as I mentioned, this is a proximity ligation assay where each spot is a foci of interaction between Oct4 and YAP1. In the presence of two of our drugs, all the spots are gone. This is an inactive compound with a similar structure, but there is no disruption here. This is a control experiment where YAP binds to other factors other than Oct4. When you put our compound, there's very little reduction. There can still a lot of interaction between OCT4 and TEAD. The control compound has no effect, suggesting that again, this is a selective disruption of the interaction. This is a chromatin immunoprecipitation assay where we show that our compounds can reduce the amount of YAP1 found on the SOX2 promoter uh, in two different experiments. SOX2 promoter, hexokinase 2 promoter, whereas there's hardly any change on a control promoter here, suggesting that these compounds can kick off YAP1 from the SOX2 promoter, leading to its suppression. So do these compounds reduce the self-renewal of cancer stem cells? Yes, it does. Even at low doses like around 2 micromolar, you can find that these compounds are preventing the self-renewal of the cancer stem-like cell. This is another compound as well. This is an inactive compound, and this is one of the classic chemotherapy drug cisplatin that is given to lung cancer patients. So these cannot eliminate the stem-like cells, whereas our drugs, which suppress SOX2 expression via YAP1, can eliminate the cancer stem-like cells. Not only that, these compounds focus on this one, they can essentially eliminate the proliferation of the regular bulk tumor cells as well at 5 micromolar, 7 micromolar, 4 micromolar, 3 micromolar doses here. And this is another cell line where you get pretty good suppression at 5 and 3 micromolar. So they can not only eliminate stem-like cells, they can also eliminate non-stem-like cells, probably through regulating some of the metabolic genes, which I'm not going to talk about in this talk. So, as I mentioned earlier, development of drug resistance is one of the major problems. There are expression of multi-drug resistance proteins and drug detoxifying enzymes in the cancer cells, especially the cancer stem-like cells. The cancer cells show amplification of drug target genes or compensatory bypass genes. There are so many mechanisms by which the cancer cells develop resistance. So, overcoming resistance and eliminating cancer stem cells is absolutely necessary. So, we examine that. Can some of our compounds eliminate the self-renewal? Yes, they can, as we said. And what happens to cisplatin insensitive or cisplatin resistant cells? So this is an experiment where we took the cells, the spheres, the stem-like cells, grew them, they grow very nice spheres. In the presence of cisplatin also, they grow very well because they're insensitive. So we took this particular cisplatin insensitive cells, dissociated them, put it back in culture, in 3D culture, in stem cell selective conditions. And what we are finding is that our compounds, the YAP inhibitors, can completely eliminate these cisplatin resistant cells, whereas these dissociated cells put back in cisplatin grow very strongly, they come back very robustly, suggesting that our compounds can eliminate the growth of the stem-like cells and especially the drug-resistant stem-like cells. That's also shown here. These are cell lines, a resistant cell line and a sensitive cell line, which are resistant to erlotinib and gefitinib. These are two EGFR inhibitors. Our drugs can eliminate both the resistant and the sensitive cells. So eliminating the drug-resistant cells is a feature of these particular cells, this particular compound. As I mentioned, the tumors contain multiple cell types, cancer-associated fibroblasts, angiogenic cell, endothelial cells, immune cells, etc. So we wanted to check how this normal cancer-associated fibroblasts, how do they respond to these cancer cells or to these drugs compared to the cancer cells. So what Namrata did is she did a co-culture experiment. She took a cancer cells, labeled them with cytotracker red color. We took green and the cancer-associated fibroblasts, which are normal-like cells. She stained them with the cytotracker green, co-cultured them, and checks what happened with the drugs. What we are finding is that 24 hours after post-treatment, these are again control cells. I mean, you can see there's a significant amount of cancer stem cell growth when they are not treated with the drugs. You get the red cell, the cancer cells taking over the culture. But these are very old compounds. These are not under the recent compounds. But when you treat with our drugs, after 24 hours and after 48 hours, you can see that the cancer-associated fibroblasts are still surviving, but the cancer cells are being eliminated. So this also shows that these drugs are selective to the cancer cells, and it also shows that they are less active or less potent against the normal cells, suggesting that there will be less cytotoxicity in the patients. Namrata also did some of the very nice 3D co-culture studies where she took the cancer cells, cancer-associated fibroblasts, and she was successful in reducing the growth of the cancer cells compared to the fibroblasts itself, suggesting that again, proving that again, these drugs essentially target the cancer cells. We also looked at cancer organoids. So these are derived from patients, lung cancer patients. They take the lung cancer, grow them as small organoids in culture, in 3D cultures. So they have not only the cancer cells, cancer-associated fibroblasts, etc., etc. They're the most accurate representation of a cancer that you can grow in a culture. And what we find is that this was work done by Jaya in the lab. Um, the red cells or the dying cells, the green cells are the live cells. When you treat with, this is a control compound, this is an active compound. What we are finding is that the active compounds that prevent the binding of YAP1 to OCT4, thus suppress SOX2 expression, they can significantly eliminate the growth of these organoids. So essentially, what we are showing is that the PPXY mimetics we identified along with the Nick Lawrence lab, they can prevent OCT4-YAP1 interaction. They prevent the self-renewal of cancer stem cells and proliferation of cancer cells with high potency. They prevent expression of YAP1 targets like SOX2, hexokinase to PDK2. They suppress adherence-independent growth of cancer cells and organoids. And they can eliminate drug-resistant cancer cells harboring either Kras or EGFR mutations. So this project is under progress. The caveat in this drug, the problem with this drugs is that so far these are very early compounds. Just because they can do this in vitro does not mean that they'll be fully active in vivo. The drugs have to be stable in the body. They should not be metabolized or excreted very fast. They should be stable enough. They should be cell-penetrant. So there are a lot of PK, pharmacokinetics, pharmacodynamic studies that needs to be done before this can be translated into even animal studies and humans. So those studies are going on, and hopefully these very early compounds will be reaching the clinic in the next 4 to 5 years if they succeed. So we started looking at other alternate mechanisms to downregulate YAP1. And I will just show in the next 3 or 4 slides, or I have another 10 minutes. I mean, in 4 or 5 slides, another pathway we are looking at. So this is a kinase, tank binding kinase. It is a non-canonical IKK. It essentially mediates signaling from the toll-like receptors in B cells and T cells. They are thought to be very important for the innate immunity. It is involved in Kras-mediated cell transformation. The reason why we started looking at this kinase is that we found that this kinase is induced by nicotine as well as EGF. And when you remove a signaling protein called beta-arrestin, that is gone. That's a protein we are studying in the lab as well. The activation of TBK1 occurs. And Smith-Apple in the lab found that phospho-TBK1 is localized on the centrosomes in multiple cell lines. So you can see that during prophase, pro-metaphase, metaphase, the staining of TBK1 on the centrosomes is significantly enhanced, and you can find it in the mid-bodies and multiple cells. It essentially very much is necessary for the mitotic progression. We also found that TBK1 could physically interact and phosphorylate YAP1. So this is again a proximity ligation as a showing. Each spot is showing an interaction of YAP1 with TBK1 with the 2 antibodies. This is another methyl transferase I'll talk about later in a minute. Single antibodies do not show any interaction. So this is again an immunoprecipitation Western blot showing that TBK1 binds to YAP1 very well, and it can phosphorylate YAP1 in vitro. So surprisingly, this is work of a fairly recent research scientist in the lab, Vishwarup Saha. What Vishwarup found was that when you deplete YAP1 protein in cells, in Kras mutant non-small cell lung cancer cells. So here you can see TBK1 is depleted. There is a huge induction in the YAP1 protein. So there are many kinase inhibitors, many inhibitors available for TBK1, but they have generally been inefficient in suppressing tumor growth, even though they are supposed to affect the NF-kappa pathway, but they were not fully effective in suppressing cancer growth. And we believe that this could be one of the reasons by which when you inhibit or deplete TBK1, YAP1 goes up, so negating the effect of the inhibitor. Again, this is showing again the whole pathway, the YAP pathway, we find the YAP1 goes up when you deplete TBK1, whereas there's hardly any change in the hippo signaling pathway component, the MST kinase, LATS kinase is happening independent of this classic hippo pathway. And surprisingly, we found that you deplete TBK1 in EGFR mutant cells. This is another gene that is mutated in non-small cell lung cancer. There is hardly any change in the YAP1 protein, whereas TBK1 is regulated. So we decided to examine how this happens. And we found that just like HIF1 alpha, YAP1 is methylated on arginine. So this is an immunoprecipitation Western blot experiment. What we are finding is that when you do an immunoprecipitation with the methyl arginine antibody, you can pull down YAP1 or showing that YAP1 is methylated, and this is elevated when TBK1 is depleted. And when you use an inhibitor of the methyl transferase, that's gone. And when you deplete YAP1, deplete TBK1, you find that YAP1 levels go up. Whereas when you put in a methyl transferase inhibitor or inhibitor of this particular methyl transferase CAR1 or PRMT4, there is no methylation. So we found that essentially this is happening through this particular arginine methyl transferase CAR1. So this is showing an interaction of CAR1 and CAR1 and YAP1 when normal cells, when TBK1 is depleted. And we found that YAP1 can be methylated on arginines by this CAR1 protein. So this is again an in vitro methylation assay. This is a histone is a control. YAP1 is methylated by this particular methyl transferase. And when you deplete TBK1, again, more YAP1. When you deplete TBK1 and deplete CAR1, the YAP1 is reduced, showing that this is happening in a CAR1 dependent fashion. And we also find that in tumors, the levels of the physical interaction of CAR1 and YAP1 is elevated. And here is an experiment showing that a CAR1 inhibitor can completely eliminate the YAP1 protein in the cell. So the reason why we are focusing on CAR1 and this particular methyl transferase is that there are several drugs that are very close to being approved in the these drugs which are arginine methyl transferase inhibitors. So these are much more advanced than the drugs we are developing, but that has its own benefits compared to this one, because this one can target multiple proteins in the cell because CAR1 has many targets in the cell as well. So this is a multispectral analysis. So this is a sequential immunofluorescence experiment that Moffitt has done for us on a tissue microarray. So this is one core of a cancer. We stain sequentially with, this is a combined staining. This is staining for CAR1, YAP1, SOX2, PRMT5, PCK1, the pan-cytokeratin. These are happening in the same tumor cells, the same tumor tissues we are looking. And what we are finding is that wherever there is higher levels of CAR1 in the tumor, there is high level of YAP1. So there is a direct correlation between the CAR1 levels and the YAP1 levels. Whereas SOX2, as we can expect, it is present only in the cancer stem-like cells. And as I mentioned, only 1% of the tumor is probably cancer stem-like cells. And you can see SOX2 going up in many of the cells. Again, every single cell where SOX2 is up, you find higher levels of YAP1. But all the cells do not show SOX2 expression because it is not a stem-like cell. So showing that, again, what we are finding in vitro is happening in the tumors as well. So this is another example of an experiment we did on cancer organoids. We are treating these organoids with the CAR1 inhibitor, SOX2 levels. These are controlled treated DMS of the vehicle. These are YAP1 levels. DAPI, you find a good amount of the cells expressing SOX2 and YAP1. When you treat with the CAR1 inhibitor, you can see that the overall levels of SOX2 going down, overall levels of YAP1 going down, and the organoids are much smaller than the control organoids, suggesting that targeting CAR1 can significantly reduce the growth of the organoids. So essentially, I haven't shown a whole amount of data, but what we are showing is that CAR1 inhibitors significantly reduce cancer cell viability with high potency. They can prevent self-renewal of cancer stem cells. I didn't show that data, but we have very nice data, dosage effects, etc. They can suppress the adherence independent of growth of cancer cells. They can prevent the expression of YAP1 target genes and gene products, including SOX2. And they can eliminate drug-resistant cancer cells harboring either Kras of or EGFR mutation. Those are some studies that are going on in the lab, but it looks very promising. And these drugs probably we believe will be very good in targeting YAP1 as well. So essentially, we are talking of YAP1, which is essential, which cannot be targeted by small molecules, small selective molecules. We have some molecules that can do that. We have pathways that can regulate YAP1, and we find that that is a highly promising strategy to combat some of these cancers. There are certain compounds out there like verteporfin or visudine, which can target YAP1, but they are highly, highly non-specific. They're cross-link all the proteins in the cells, and they're highly light sensitive. So we believe that these class of compounds, the OCT4 YAP1 disruptors we identified, as well as these hormone inhibitors or the methyltransferase inhibitors, could be very good molecules to target YAP1 and reduce stem cells and cancer progression and metastasis. So I guess this is I have 1 minute left. So these are the people who did the work. This is one of our lab lunches a year ago. Mohan Kumar and Namrata, they did all the work on the stem cells as well as the drug discovery effects. Vishwarup Saha did the work on the hormone inhibitors. Jaya and others contributed to multiple projects in the lab. Smita Pillai and Piali Das Gupta's Courtney and Deepak and Sandeep Singh, they all contributed to the early parts of the projects I mentioned. We have many, many collaborators without whom these projects will be possible. Nick Lawrence and Sujiva are chemists who contributed significantly. Domenico Coppola is a pathologist, and these are many other people whom we work with, and we have been blessed to have funding from multiple sources. With your permission, I will stop here and answer any questions you may have.
Yeah, thank you so much, sir, for your presentation. It is really a thought-provoking session for cancer aspirants as well as for many life sciences aspirants. Thank you so much for your presentation, sir.
You're welcome, Papa. You're welcome.
Yeah, with your permission, I have few questions for you, sir, from your presentation today. If you permit me, I will
Sure, please.
forward one by one to you.
Go ahead.
Yes. Yeah, thank you, sir. The first question to you is, we come across cancer-like stem cell and non-cancer-like stem cells. How do you people identify what is the strategies used to identify cancer-like stem cells with that of non-cancer-like stem cells?
So essentially, cancer stem cells arise from mutations occurring in non-stem cells. So there are adult stem cells in each organ. Lung has its own adult stem cells, which essentially promote the regeneration of the lungs if the lung gets a injury. Liver has its own adult stem cells sitting around. So the difference is that they do not form tumors or anything like that. They do not harbor many of these markers what I mentioned about. So usually you do not find them in a regular, say, in a cancer setting. So if the point is, can these drugs affect the normal adult stem cells? They may, but it is highly unlikely because we'll be targeting the tumors predominantly, and the prevalence of the cancer stem cells are much higher than the prevalence of the adult stem cells, and the access of these drugs to the adult stem cells is little bit reduced. So some of the markers, like for example, the transcription factors, what we talked about, the OCT4, SOX2, nano, KLF1, but using CRISPR-Cas9 to eliminate or to delete the YAP1 promoter, it works, but I don't know how you can use it as a therapy or as a therapeutic function. You can do that in cell line. So we have eliminated YAP1 protein itself in the cell. You don't have to essentially eliminate the promoter region. You can eliminate the first exon or the first exon and second exon junctions, and you can essentially eliminate the whole YAP1 protein. You don't have to necessarily target the promoter region. But how can you do that in a selective cancer cell specific fashion to give it as a therapeutic agent is a question. Eventually, you may be able to do that. You may be able to express a specific sgRNA under the control of these promoters, which are expressed only in cancer cells. Except those are possible, but those are probably few years down the road.
The next question that to you is, we can from your research, we could understand that there are some subset of stem-like cancer cells which forms the vascular network. Do you analyze any molecular mechanisms through which those subset of cancer cells produces the vascular network?
So we don't have, nobody has too much mechanistic aspects on that one of how it happens. Other than that, we know it is the stem-like cells that are doing that, and the secretion of VHF by the adjacent cells or the close by cancer cells promote the formation of these, but not too much biochemistry is known about that. Unfortunately, there is not too much literature on that, how it happens.
Yes. Okay, so I'll pass on to my next question. When it we come to a state of hypoxia, there we come across proline hydroxylase, where it has been can we suppress the proline hydroxylase so that we can overcome the hypoxia? Is there any molecular mechanism used to
So,
suppress
so,
proline hydroxylase activity there?
so
So
there
that
are,
we can stop the transcription.
so you cannot, prolyl hydroxylase does not work at the level of transcription. It works at the level of the protein stability, again, protein degradation. There are very specific inhibitors of prolyl hydroxylase 2, which is a main hydroxylase that hydroxylates the prolines under normoxia conditions. So under normoxia conditions, as I mentioned, prolyl hydroxylase actually hydroxylates HIF1 alpha, YAP1, which are these oncogenic proteins, and keep their levels down. So if you essentially inhibit the prolyl hydroxylase, these protein levels will go up. So from a cancer point of view, that will be detrimental. You are essentially promoting the growth of such tumors. So inhibiting prolyl hydroxylase 2 may not be a best strategy to combat cancer. But such a strategy will have benefit in other situations. For example, wound healing. There are people who get wounds in war as well as in accidents, etc., whose wounds take a long time to heal because angiogenesis is slow, etc. In situations like that, a prolyl hydroxylase 2 inhibitor may elevate the levels of HIF1 alpha, elevate the levels of VEGF, and promote the wound healing. So there are situations where a PhD inhibitor will be useful, but not as a cancer therapeutic agent, to the best of my knowledge.
Sure. So my next question to you is coming to SR6 compound. How this compound targets YAP1 and SOC2? What is the mechanism behind in targeting these compounds in targeting the YAP1 and XOX2 level?
So these compounds were developed in a very sequential fashion. So we started with a compound that was not very active, started with the multiple compounds that are not very active. Then we optimized it in an in vitro screening assay. I didn't talk about the screening assay very much because those studies were funded by industry, and we cannot disclose all the how it was done. So we optimized the compound to get a very high potency. So this compound essentially prevents the binding of OCT4 to YAP1, perhaps by binding to the YAP1 protein. So these compounds will bind to a region of YAP1 or associated with YAP1 and preventing it from binding to OCT4. And YAP1 has to bind to OCT4 to induce SOX2. So in our assays, first we did the binding assay to find that it disrupts the binding of OCT4 to YAP1. And then we did the assay to check whether it binds to OCT4 and sequential assay and another 500 compounds what we synthesized at Moffitt in our cancer center. So that is how we reached those compounds.
Yes. Uh, Now I have some few questions from our audience, so that with your permission, I will take those
Yeah.
questions. Sure. Uh, the question is from Goshwani, and she asks that how can we develop alternate biochemical models to combat cancer like stem cells?
So there have been many strategies to combat cancer stem-like cells. So the hippo signaling pathway and the YAP1 I mentioned. The hedgehog pathway is another pathway that's involved in now cancer stem cells. There are Wnt signaling. So these kind of, that's a very good question, because what it shows is that you should have a good understanding of the biology of the basic science of cancer stem cells before you can develop a good therapeutic target. So once you understand which are the main pathways that are involved in promoting the growth and self-renewal of the cancer stem cells, you can develop inhibitors for them. Some of the cancer stem cells we study are highly sensitive to inhibitors of EGFR, for example, epidermal growth factor receptor. So once you know which are the pathways involved in a specific cancer stem cell or specific type of cancer stem cells, you can target that. I should also mention the biology of cancer stem cells or the main drivers of cancer stem cells are different in different types of tumors. A drug that may work for the lung cancer stem cell may not work in, for example, in a breast cancer stem cells, and vice versa. So one should understand the biology of this cancer stem cell, and then based on the signaling pathways that drive the stemness, you can develop small molecule inhibitors. So that is where the basic science, which many of us are doing, is very interesting. I mean, and very important. You cannot develop a drug, you cannot develop a translational product unless you do a very thorough biological study, basic science study. Without good basic science, you cannot translate or develop a drug. So that's why basic science is so important, and which people seem to ignore that fact now.
The next question is that Srikumar Chellappan, and he is asking that is there any strategy by which we can convert the stem-like cancerous cells into non-stem-like cancerous cells using non-cancerous stem-like cells since they aren't harmful?
So essentially, it is very easy to do that in an experimental system, but it may not be that easy to do that in a actual cancer patient or in a mouse model. So in the lab, in the experiment, so you isolate the stem-like cells, and unless you grow under very specific conditions, you have to grow them on low adherence plate in stem cell selective media. Those are the conditions needed to maintain the stemness. So if you isolate the stem-like cells, put it on a regular tissue culture dish, put it in the regular cell culture media, they differentiate into non-cancer stem-like cells based on the growth factors, cytokines. So they become the non-stem cells very, very easily in an experimental system. But in vivo, in the presence of the tumor microenvironment, it is not very easy to convert them into a differentiated cell. It will be much more easier to find the signaling pathways and eliminate the stem-like cells. I hope I answered your question.
Yeah, sure, sir. Shall pass on to the next question. And this question is by Vishal Kumar Shagu, and he asks that you have shown us that DMSO also alters the expression of YAP1. So during freezing of cells, we are actually affecting the natural behavior of the cancer cells. Should we change the freezing agents?
No, you should not. I mean, I didn't show that DMSO changes the expression. DMSO was our control. That is our baseline. Because many of the drugs we used were initially dissolved in 10% DMSO. So we used 10% DMSO as a vehicle. I don't think in any of the experiments we showed that 10% DMSO really alters the expression of these proteins. So DMSO, when you freeze cells, that is absolutely necessary to make sure that the cells are frozen in a uniform fashion. And that is absolutely critical. So please continue to do that. I mean, that is absolutely essential. Without DMSO, your recovery rate will be quite low unless you use a substitute like other substitutes out in the market. So that is our vehicle. That is our control solution.
The next question is from Ashita, and she asked that I would like to know what if the proposed molecule to be developed in a drug is not efficient in penetrating a tumor cell? Is there any backup plan in your mind?
Can you repeat that question once again?
Yeah, sure, sir. I would like to know what if the proposed molecule to be developed into a drug is not sufficient in penetrating a tumor cell?
So that is a very good question. So if I remember, if I mentioned, first we have an in vitro assay that is all done in a test tube where we find that the compounds prevent the binding of OCT to YAP1. We do it at the peptide level that do that. And only we find only a small percentage of those initial compounds will work in our next assay, which is looking at SOX2 expression. To a great extent, it is because many of these compounds do not penetrate the cells. So we test a large number of compounds and filter down the active compounds which can penetrate the cells and can suppress SOX2 in a specific fashion, which can kill the cells in a specific fashion. And improve the efficacy, the stability, as well as self-penetrance of these compounds. So it is a constant progress. So we have a compound 52, 53, etc., which seems to be highly potent so far, but we are trying to even still improve those compounds to enhance their cell penetrance, their efficacy, their cell kill, etc. So it is a sequential, you have to go in multiple circles. You take this compound, analyze it, see what is wrong or what is happening, then go back again, try to modify it. So it's like a constant progression of what is happening.
Yeah, sure, sir. The next question is from Damsha, and he is asking that, please, if we are able to uniquely study cancer from a patient, can we identify a specific mutation that caused it? And if is there or any way we can reverse a mutation?
So you can, so now I think it is a standard of care in most of the good hospitals in India as well. If someone goes with a lung cancer, for example, they will do a targeted sequencing for the main genes that are mutated in lung cancer, for example, EGFR gene, Kras gene. This whole panel, they will run a sequencing to see which genes are mutated. So in India also, most of the hospitals that do c-met inhibitors, SRC inhibitors. So based on the mutations, you can use small molecule to treat the patient. And another thing, don't forget, the immunotherapy is another big thing that's going on now, which has been highly successful in around 35% of lung cancer patients. So you can do those kind of things based on the sequencing. For example, they look at levels of PD1, PDL1, um, certain genes like that, and see whether the patient will respond to immunotherapy. You cannot go back and correct the tumor, but you can combat the molecules and prescribe the appropriate therapy based on the mutations. That is what is being done.
Yeah, the next question is from Shubhash's coach, and he is asking that why vertiporin is insufficient for targeting YAP1.
So verteporfin, it was identified in 2012 in a large scale screen. In vitro experiments, it is very efficacious. The problems with verteporfin is that it is light sensitive and it is highly toxic. Um, so people are trying to do it and use it in animal experiments, and there are proposals to use it in clinical trials. And I think it has been approved in clinical trials, but the patient has to be kept covered in dark clothes for 3 days or once the drug is given. So it is highly toxic and it is highly non-specific. As I mentioned, YAP1 is not the only target of verteporfin. There was this very nice paper that came out 3-4 years ago that showed that verteporfin can cross-link a huge variety of proteins in the cell. And if you they did a Western blot or they did a protein gel of treated cell, untreated cell, and showed that the verteporfin treated cells, they were all migrating as a group much slower than the other ones. Everything is cross-linked. So it can has severe cytotoxicity as well. So that's why it is not a good drug.
Yeah, the next question is that can some cancers caused by stem cells classes be eradicated through antibodies?
So the cancer stem cell theory is that most of the cancers are caused by cancer stem cell. They are the ones that initiate and propagate the tumors. So the antibodies, for example, there is a lot of immunotherapeutic agents, as I mentioned, ipilimumab, nivolumab, which are anti-PD1, anti-PDL1, what they call as the immune checkpoint inhibitors, which used to be highly efficient in lung cancers and melanomas, which are essentially very high in mutations. So they are high mutation-rich tumors. So those kind of tumors respond very well to immunotherapy. That mainly works independent whether it came from one particular type of stem cell or not. It just depends on what protein is expressed on the cell surface. For example, most of the tumors that respond to the PD-1 inhibitors will have PD-1 gene or PDL1 gene expressed on the tumors and PD-1 expressed on the tumor cells, on the T cells. If so, they do an analysis for PDL1 on the cancer cells. If the patient has high levels of PDL1, they give them the immunotherapy that targets the PDL1 or the PD-1, and the patient responds most of the time. But if the patient has low level of this particular immune marker on the tumor cell, they won't respond. So that is irrespective of whether the tumor came from, where the tumor came from, it's a specific stem cell or not. The immunotherapy response is very different.
Sure, sir. Shall pass on to the next question. The next question is again from Subhash Ghosh, and he asks that can you discuss about the crosstalk between node signaling and YAP1 signaling in cancer stem cell contest? What can be the putative strategy to target the both?
Which was the hippo signaling and which was the other signaling? What you mentioned?
Node signaling and YAP1 signaling.
So actually, that's a very good question. I mean, there are our lab does not work on notch signaling, but there is a significant effort going on to target notch as well as wind and similar signaling pathways. So there are a lot of inhibitors being developed, especially in the wind signaling pathway and to a certain extent notch signaling pathways. But as you know, they are also very active in many of the normal cells, especially during development, as well as some of the adult stem cells, etc. But those work is still going on, and they play a role in cancer stem cells. But again, depending on the cancer type, so you cannot use the same inhibitors or target the same pathway in one tumor versus the rest. For the YAP1 and the hippo pathway, the major challenge, the wind and the notch, they essentially promote growth. Those are oncogenic pathways for the most part. The hippo signaling pathway, the major challenge is that it's a tumor suppressive pathway. The only oncogenic components are the YAP1 and the TAS. Because when the cells are loosely divided, when the cells are tightly packed, this pathway essentially prevents cell. When the pathway is active, cell proliferation is inhibited. So you cannot find an inhibitor, for example, the LATS1, MST1, etc., etc. So you have to specifically target YAP1 or TAS protein, which are the only oncogenic component of this tumor suppressive pathway. So that is the challenge with the hippo pathway. So I am sure that the wind and the notch pathway inhibitors are more likely to be in the market and are fairly soon, especially the beta-catenin, the wind beta-catenin pathway. Even beta-catenin is not that easy to target, but those will be fairly useful and more widespread in the coming years.
The next question is that does cancer pass from one generation to another? And is there any mechanism or clue so that we could predict or suspect that this person might be infected with the cancer?
Yeah, there is a huge amount of literature on that. Many of the cancers can be genetic, especially breast cancer, ovarian cancer, things like that. Many cancers have a genetic component, BRCA1 mutations, BRCA2 mutations. Yes, they encourage screening if the people's mothers or aunts, or if there are family members who have breast cancer and ovarian cancer driven by these particular mutations, people can get screened and they can be more vigilant, they can be undergo screening more often, etc. So there is a huge amount of literature on that. I suggest you to do a quick Google search and find that. I mean, there's a lot of very simple, very nice articles on cancer screening and how it can be eliminated, how it can be controlled. So one of the reasons why, as I mentioned, the incidence of cancer has gone down is mainly because of such vigilance, because of the education, the smoking cessation, the early detection, etc., etc. So that field has grown so much, and it is making such a big difference in the incidence of cancer. Yes, it's very possible.
Sure, sir. I'll pass on to the next question. The next question is that how bioinformatics tools and molecular docking and dynamic stimulation helps us in our battle with cancer.
All those are very important. I mean, the docking experiments and people do to understand the structures of the proteins and to develop better drugs. I mean, such molecular modeling studies are very useful during drug discovery and drug development. Those things were not possible, for example, 20-25 years ago, but those things have become much more easily doable now. Even computational modeling of proteins and drug the screening based on the modeling studies, those things are highly possible and they speed up the drug discovery process very well if the protein structures are known. Actually, artificial intelligence studies, the gene expression bioinformatics studies, there's a huge amount of such informatics studies that are taking place to combat cancer, to find what combinations of mutations, what gene expression patterns can be targeted by what drug. And another huge thing, especially here at Moffitt Cancer Center, is the mathematical oncology. We have a whole department on mathematical oncology, in addition to a machine learning department. So the mathematical oncology department, what they do is they model cancer growth and model response to therapy. So they, based on their experience, then they can take cancer cells from patients, they grow side by side, and they can predict based on the response that this particular drug regimen will be beneficial to this patient, and this is how you should give. For example, one of the classic ways of doing cancer therapy is you give a huge dose of chemotherapy drugs, eliminate the cancers, eliminate the cancers. So what people have found is that when you give a huge amount of drug to a patient, as I showed in my talk, it is a differentiated cells that are responding to the drugs, and they are the ones that are killing away. They are being killed away by the drugs. So the only surviving cells are the cancer stem cells, which will sit around for some time and come back. So one of the major things what they're doing by the mathematical oncology people are doing are to give the dosing regimen such that you reduce the dosage amount so that you don't completely eliminate the cancer cell. You keep cancer as a chronic disease and enhance the survival of the patient till you reach a stage where it can be removed by radiation or surgery or stuff like that. So there are all this information, the math oncology modeling, the artificial intelligence, the machine learning capabilities, the bioinformatics are playing a whole new role in the development of cancer therapeutics.
Yeah, thank you so much, sir. The next question is, how do you see your cancer research being and with that of in abroad?
There are fantastic labs in India that do very good cancer. I mean, whom I personally, Manish Kumar, but Gopal Kundu, Tapas Kundu, and there are whole scientists. Sandeep Singh, who is tomorrow. I mean, there are so many fantastic cancer research in many institutions. IAC, Satish Raghavan, and many other people. So Kumar Somsundaram. There are actually the type of studies they are doing in India are very exciting, very good, and comparable to many cancer institutions here. I mean, there are some challenges there, but the standards of the science that is being done in India now is vastly different from what it was 10 or 15 years ago. I mean, it is remarkable what is going on there. I am highly enthusiastic and very happy to see how things are changing for the good there, especially the use of informatics and things like that that Indian government and the Indian institutions are doing is very cost efficient, and it is remarkable. As I mentioned, now there's a Indian Cancer Registry that they started 15 years ago. Now they are capturing huge amount of information on the cancer incident, what type of cancer, what is the age of the patients. You can get a huge amount of information that was just not gathered till 10, 15 years ago. So things in India are extremely good, actually. I mean, there are fantastic labs there.
Really happy to listen this from you. And what would be the take-home message for all our audience today from you?
So the basic message is that cancer is not one disease. It is very different from patient to patient, tumor type to tumor type. It requires a huge amount of work, understanding of the basic biology of the disease to devise good strategies to combat cancer. But we will get there. As I mentioned, the cancer incidence is reducing. The response rates are significantly better than what it was 10 years ago even. So we should continue to work to understand the biology of the cancer, develop new therapeutics, and use the information that is available. I mean, what type of people are getting what type of cancer. As I mentioned, for example, in Northeast, there are certain areas, certain districts with very high incident rates of cancer compared to anywhere else in India. We should conduct such population-based studies as well to understand the etiology of this cancer. Why is this happening? So we should conduct the basic studies and try to base our drug development strategies based on the actual science, and that will improve the outcome significantly in the coming years. That's all I had to say.
Yeah, thank you so much, sir. And before you leave, this might be a last question for you. And the question is, what are the ways to get eligible if a person willing to pursue his or her research in your laboratory in USA from India?
So in India, so as you saw from my acknowledgement list, I had fantastic postdoctoral scientists from India. I mean, all these years I had some, I mean, very outstanding, very nice people. And so if you do a very good PhD with good training, with good papers, you are more than welcome to apply here to not only my lab, to any other lab in Moffitt. I mean, so we are all looking for good postdoctoral fellows. The important is to have good publications in international journals. If you have made good presentations in international conference with your biology, with your posters, if you have good training, if you have a good interest in cancer biology, please do apply. And yeah, we all look forward to. We also take PhD students in our international students in our PhD program. We take around 15 students every year, and you can find the information on the web. Out of which we have taken 2 or 3 students from India pretty much the last 2 or 3 years. So they are also for the PhD, for the apply to the PhD program. You need to have the GRE. You should have some research experience there. I mean, either during your masters or after your masters, especially in the cancer or eukaryotic or animal field. You should have some experience, and your science should be good. You should have a good theoretical knowledge and good grades and everything. And yeah, we take, I mean, our postdocs are as good as anybody you find in the US, I can tell you that. So so we are all looking for all that.
Yeah, thank you so much, sir. This will be helpful for many participants in our event. Thank you so much, and I thank you for your accepting my invitation and listening patiently in all our questions and answering our questions. Thank you so much, sir.
Thank you, Partha.
And
I really enjoyed it. Have a great night, and congrats to India on the cricket win yesterday. I watched most of it, so it was very
it was also a thought-provoking session. Thank you so much.
good. Bye. Good night then.
Thank you. Bye bye.
Bye.